Good morning. I'd like to thank the organizers of this meeting for giving me the opportunity to come here and present to you some of our recent findings that we made at Rutgers University during the last 6 years in collaboration with my students. My title is the Planckian information theory as a unified theory of organizations in physics, genomics, neuroscience, glottometrics, econophysics, and cosmology. My talk will consist of mainly 3 parts. First, I'll present to you the observations with little or no explanations. And then I'll summarize some patterns we've seen among these observations. And then we'll present some of our tentative explanations for these observations. Our observation starts from blackbody radiation phenomena. As you all know, the blackbody spectrum, radiation spectrum looks like this. And the Planck's equation fits the spectra perfectly. And our equation, which is derived from Planck's equation as given here, just by replacing these constants in Planck's equation with 3 parameters, A and D, initially, and that was enough to fit the blackbody radiation equation exactly with this equation, of course, because these are all parameters or constants. Later on, with the help of one of my students, Kenneth Saw, we expanded the equation by adding 2 more free parameters, capital A and capital B. And with these expanded equation, we could fit just about any fat-tailed histograms. So far, we've fitted about 20 of those coming from all fields in science and humanities, including physics, atomic physics, and economics, and cosmology. And this is the observation that I will show you in about half a dozen slides. First, the first observation was that people in 1997 isolated cytochrome oxidase with flavin at the active site and measured the turnover time of the single enzyme. As it turns over, the flavin fluoresces and fluorescence disappears. So it's a blinking phenomena. So by measuring the time for this fluorescence to last, they could estimate the turnover time of this enzyme, which is a catalyzed chemical reaction converting cholesterol to cholesterol. And this blue line is their histogram of the turnover times of a single enzyme. As you can see, single enzyme catalyzed chemical reaction with different turnover times at different times. And that histogram reminded me of the blackbody radiation equation that I learned in grad school 30, 40 years ago. So I used the equation to fit the blue observation, and this pink is my hand fitted curve there. As you can see, it seems to fit fairly well, but later on the fit was better when I used the solver program in Excel to do it more automatically. And yeah, that may appear later on. Now, this is simply a repeat of fitting our equation to the blackbody radiation spectrum. Okay, and this is the equation fitting to the free energy of protein folding. As denatured protein folds into native form, there's a heat produced, and people were able to measure the heat and entropy changes. Also measured, was able to calculate free energy changes accompanying protein folding, and that's called folding free energy. And that's plotted on the x-axis, and the free y-axis is the number of proteins out of 4,300 that fall in given free energy. So this histogram also fit our equation, given in red. This is a receptor on a T cell, and this portion of the protein has a varied nucleotide sequence. So the gene coding for this part of the receptor, T cell receptor, the probability of the various sequences appearing in the gene were estimated by people at Princeton, and their predicted generation probability is given in blue, and our equation called Planckian distribution fits that distribution perfect. This is this is a repeat of the enzyme I showed you earlier, and this is how the solver fits the observation, which is much better than my visual fitting that I showed you first. This slide shows the RNA levels inside the budding yeast. When you perturb the cell by switching the nutrient from glucose to say galactose, RNA levels inside the cell changes in a growth genome-wide manner, about 6,300 of them, and you can make a histogram out of the free energy and messenger RNA changes occurring in the cell, measured by microarray technique. And the histogram from that measurement is given in the blue. And red is the Planckian distribution, and green is Gaussian distribution. And Menzenerath-Altmann is the equation that linguists have discovered that fit the word length frequencies or sentence length frequencies in text. And this equation is completely derived from empiricism, and they also fit the same data that Planckian distribution fits, indicating that one physics can be fitted by more than one equation. This is the same RNA measurements, this time made in human breast cancer tissues, and experimental and Planckian. This is a human RNA levels from human breast cancer tissues from 20 patients. This is the Planckian equation, a 4-parameter version. And when you fit this equation into different histograms, you can characterize the shape of the histogram in terms of 4 parameters: A, small b, capital A, capital B. And these parameters, especially B over A, seems to be sensitive to the structure underlying the histogram, the data. This is applied to human breast cancer tissue RNA levels, and the human genome has hundreds of different metabolic pathways, and we looked at 6 different pathways, and all these 6 different pathways have different B over A ratio. As you can see, these are p-values which are all smaller than 0.05, indicating that this equation can differentiate this pathway from that, this pathway from that, and so on. So all these 6 different pathways can be differentiated by the numerical values of this equation, indicating that this equation not only fits to many different data sets, but within the data set, they can differentiate subgroups in it. And this table indicates the RNA level changes in the human breast cancer tissues after treating with the anti-cancer drug, doxorubicin. And this is before treating with the drug, this is after treating with the drug, and these ratios vary slightly here. But as you can see in the p-value here, this pathway detects the difference between before and after drug treatment, but these other pathways don't. So not all pathways are affected by anti-cancer drugs. This is functional magnetic resonance signals measured from live human brain. These data are all from the internet, by the way. Yeah, I didn't give the references. On the x-axis is the distance from the mean. These areas are where the interesting things are happening when you perturb the human brain. And these areas can be divided into hundreds of volume element voxels. So each region has an average intensity, and different voxels have a distance from that average distance, average intensity, and that's plotted on the x-axis, and y is the frequency. The experimental value is given here, more wavy, and the Planckian distributed fits fairly well. So, functional magnetic resonance imaging signals can be simulated by the equation. And this is the EEG measured from outside the brain. And that also fits Planckian distribution. This is a whole brain phenomenon, decision. You can measure the time it takes for a person to make a decision. And the more difficult the problem you're presented this people with, the longer it takes. So people have measured the so-called decision time on the x-axis and the frequency. Given repeated stimuli, they can measure different times, and that's made into histogram in the blue, and that fits Gaussian, Planckian distribution perfectly, and Gaussian is like this. Now, I'll come back to this. Here is an interesting histogram generated from Kerry's speech given in 2004. On the x-axis is the word length, that is the number of letters appearing in the words that he used in his speech. And this is frequency. As you can see, the observed is blue, Planckian distribution, and the Menzenerath-Altmann law. They all fit the same data. And this is not surprising because that's the law which was formulated to account for these kind of things anyway. But it is surprising that Planckian distribution also fits the same data. And this is the most recent one. This is a histogram of the annual income in the US in 1996. This is income level, this is a percent of population, and observed is blue, red is Planckian distribution. And somebody in the physics community tried to model the annual income distribution with an exponential function based on Boltzmann equation, and as you can see, it deviates. And this is what I call the ultraviolet catastrophe of the second kind. And that fits, we can avoid that by Planckian equation, and there may be some interesting principles behind this. And finally, although controversial, in July, as you all know, some scientists have claimed to have measured the polarization of the microwave background radiation from the Big Bang, and I plotted their measurement in the blue, and Planckian equation doesn't fit too well. This is the worst fit I have tried, no matter how hard I try, I couldn't fit it. And this is Gaussian, and this is experimental. And one possibility for this, now I'm getting into the explanation, but one possibility is that when data fits Gaussian distribution, we can say that the data were generated by some random processes. So when the data fits Planckian distribution, which deviates from Gaussian, we can say that there's some kind of organization going on, or non-random process going on. So the degree of non-randomness intrinsic to the Planckian distribution would be proportional to the difference between the Gaussian and Planckian. The further Planckian distribution is from the Gaussian, we say there's more information buried in the Planckian distributed data. Since the astronomical data here is in between, that means the astronomical data has less information than predicted, perhaps because of the interference by the galactic dust that they're talking about. So this figure, if true, would predict that the Planckian, the polarized microwave background radiation that people measured, for which they were criticized for various reasons, may be true 2/3 of the time, and then 1/3 of the data may result from interference by the galactic dust. And this should be testable. I have not approached the astronomers yet, but I intend to. So these are the observations. Now, let me give some regularities or some explanation. The reason that blackbody radiation derived Planckian distribution fits single molecule enzyme catalytic data, maybe because enzyme energy levels are not homogeneous, but they are discretized, just like electronic energy level in atom is. Once you have discrete energy levels inside an atom or inside an enzyme, they can fit the same equation. So the first observation we made of the Planckian distribution fitting enzyme data can be explained by this. The only difference is that in atoms, electrons are all in the lowest energy level. But in enzymes seems to exist in different ground state energy levels already.
Sung,
yes.
Could you just define what you mean by enzyme and catalyst?
This is an enzyme. Enzyme has to be activated by thermal motions to reach transition state in order for enzyme to break bonds, to catalyze.
Okay, so what are the C1 to C2?
These are different conformation. Enzymes can exist in different conformational state.
Oh.
And each conformational state may have unique free energy. And some have low energy, some have high free energy. Those who are already in the high energy, free energy state, it takes only small further input of energy to do catalysis, as compared to the enzyme, which is low energy state. Yeah.
So Peter, is this a duality?
Now, another general idea that we can import from atomic physics to other fields that fits Planckian distribution may be wave-particle duality. The Planckian distribution mathematical form is a product of two factors. This is power function, and this is exponential function. The power function times exponential function. And Planckian equation tells us that the first term is related to the number of sending waves per unit frequency per unit volume. Volume is important. The atom has very small volume. The universe has a large volume. But they might both affect this term. And this term is the average energy per standing waves. So because of these two factors in the Planckian distribution, it may be able to account for data generated from different scale system and different energy system. That is schematically shown here. So if you have any object that you are observing, whether it's an atom, enzymes, or human brain, you can treat them as a volume. When you have volume and you input energy, there may be standing waves formed, whether it's an electromagnetic wave, concentration waves, mechanical waves, and so on. And these standing waves interact to output results which we measure as blackbody radiation, protein folding, enzyme catalysis, RNA levels in cancer tissue, T-cell receptor gene diversity, functional magnetic resonance signals, or annual income in the society, decision time, word length, distribution, and I have even sentence length distribution histogram also, and cosmological radiation phenomena. Another idea suggested by one of the data that fit by Planckian equation has to do with enzyme catalysis. Enzyme catalysis is a fundamental to explaining life, but we don't have still a good model, I'm afraid. And in thinking about enzyme catalysis fitting the radiation spectrum, I think we can learn a lot from physics of laser. As you know, laser involves promoting electrons in the ground state to excited state by input photons. And when the electrons populations are inverted, you can trigger the deactivation of the excited electrons to ground state by triggering photons, so that you can amplify light. Something similar may happen inside the enzyme. Enzyme, as I said before, are existing in the different ground state free energy level, and they have to be promoted to transition state and to active state due to ground motion, heat. And at this state, just like in laser, all the enzymes in this state are deactivated to ground state upon binding substrate. And when that happens, heat that run into enzyme comes out as heat, but in addition, catalysis happens. So this is another similarity between atomic processes and enzyme processes, as I see here, which may explain why blackbody radiation equation can also fit the enzyme catalysis. And this is the further generalization of this mechanism, okay, which I call sit-tem-toff mechanism, signal-induced deactivation of thermally excited metastable state. Excited metastable state is missing here. To function. And this is simply a global generalization of this mechanism. Okay, this is identical. Categorical theorists will say they belong to the same category. So is this. So all of these are different categories belonging to the same system, and there's a connection among these, which categorical theorists will call functor. There's a functor underlying these similarities. Another mechanism suggested by decision-making data fitting Planckian distribution comes from so-called diffusion model of decision making, which has this differential equation. X is this evidence accumulated at time t. A is the evidence accumulated each time step. And C is the C determines the amount of diffusion. dW is the Wiener process or Brownian motions. Using this equation, they could fit experimentally observed decision time, okay? And so because of this term, which is random, you have Gaussian distribution there. So Gaussian distribution and Planckian distribution is coupled through this differential equation. And interestingly, you can generate Planckian distribution, the red line here, from Gaussian distribution Gaussian distribution by simply transforming the x-coordinate of the Gaussian distribution non-linearly while keeping the y-coordinate of the Gaussian distribution invariant. Then you get the blue line, and which coincides with the Planckian distribution, indicating to me that there is a Gaussian component to the Planckian distribution. Or you can say Planckian distribution is a sort of a perturbed Gaussian distribution, or asymmetrized Gaussian distribution, based on this model. So one of the conclusions that I have been led to, for which I may be criticized, is that the Planckian distribution and Gaussian distribution going together. In other words, the rising portion of the Planckian distribution is always perfectly coincident with Gaussian distribution. So if you take the difference between the two, you get an area, and that I view as a measure of information, which I call Planckian information. And this information is a measure of the order, because it's a deviation from the Gaussian random process. And this may be compared to Boltzmann-Gibbs entropy, which is a measure of sort of a disorder. So we may have a quantity here, mathematical quantity, which is opposite to Boltzmann-Gibbs entropy. This is the definition of the Planckian information. Binary logarithm of a ratio of two areas under the curve, Planckian and Gaussian. To summarize all these observations in a coherent manner, I was led to study category theory. I'm not a mathematician. I don't know much about category theory, but to the extent that I know, I think we can say the last slide that I'm going to show you. Before that, let's review what category theory is. This is comparison and analogy are fundamental aspects of knowledge acquisition. We argue that one of the reasons for the usefulness and importance of category theory is that it gives an abstract mathematical setting for analogy and comparison. I've been comparing different fields. That's what attracted me to this statement. Allowing analysis of the process of abstracting and relating new concepts. This setting is one of the most important routes for application of mathematics to scientific problems. Another statement by Brown and Porter: we view a category as giving a fairly general abstract context for comparison. They emphasize comparison a lot in their paper. The objects of study are the objects of the category. Two objects, A and B, can be compared if the set CAB is non-empty, and various arrows A to B are ways of comparing them. The composition corresponds to if we can compare A and B with C, we should be able to compare A with C. This is the easiest definition of what composition condition is of the category theory that I've encountered. A functor is a way of comparing categories. So category theory is really a mathematical ways of comparing to things that we do all the time without using mathematical language of category theory. So what is a functor? This is a statement by Spivak, with whom I've been communicating recently. Different branches of mathematics can be formalized into categories. These categories can then be connected together by functors, and the sense in which these functors provide powerful communication of ideas is that facts and theorems proven in one category can be transferred through a connecting functor to yield proofs of analogous theorems in another category. A functor is like a conductor of mathematical truth. I'm applying this statement to what I have observed. All you have to do is replace mathematics with natural or human sciences. All branches of natural human sciences can be formalized into categories, and then you can compare them. And whatever the way you can compare different branches of human knowledge, we can call that functor, according to this statement. And before showing my last slide, I just want to indicate that I had same or similar idea as mathematicians called category theory in 1991. I published this in 1991 and further elaborated in 2012. I call this table theory. I don't have time to go into detail that, but if you analyze what I call table theory, you will see the similarity to category theory. Only this is using words. Category theory is using mathematical symbols. And in here, 4 nodes and 4 arrows here, this is a commutative square. I recently found out, okay? This is a commutative square in category theory, and I didn't know that. But all I know is that this works. Okay, so this is the final table. This I have gone through already. So here is the Planckian distribution again, and here are the fields to which this can be applied. Atomic physics, cosmology, cell biology, brain physiology, society, and universe. And these are systems of components which are connected arrows. And these are called nodes in network science, and these are arrows, or these are objects in category theory, and these are morphisms in category theory. And as you can see, these connect all these different fields, hence we can regard this as a functor. But not presented any evidence for this set. I've long tried to explain my observation in terms of Persian semiotics, or sign theory by Peirce. So to make a long story short, opposite to this equation acting as a functor, there's a system of symbolic signs, symbolic text, which can also connect all these different fields, which I call functor also. The difference is this is a quantitative and this is qualitative. So I call this quantitative functor and this is qualitative functor. So these two, quality and quantity, may play an important role in organizing human knowledge, and the principle underlying the duality of quality and quantity is, I think, the complementarity. Thank you. Yes.
The Planckian distribution is for the oscillations of a oscillator, and that you can derive with Planck for a system of oscillators. In electrodynamics, you describe the atom, enzyme, cells, brain, and even the interaction of society through oscillations. And so the reason that the Planck distribution works is they're all based on oscillations. But this was learned much earlier at the beginning of the 20th century when the French proposed the structural philosophy of science, and they won many Nobel Prizes in linguistics, economics, many different fields, showing this relationship where it's structural due to the common structure and forces like electromagnetic forces. So you're seeing that with your functor. I noticed you have semiotics here.
Yes.
The semiotics is related to the structural philosophy of science that the French introduced. And
Semiology.
we have our current philosophy of science, the postmodern philosophy of science, as a reaction to that because we didn't like it because it causes us to have to throw out our quantum mechanics, our relativity theory. And so in order to preserve that, we made separate silos of science where the definition of truth is different in each silo. But what you're seeing is it's got commonality. And that's what the French were saying in their structural philosophy of science. And there's whole books on that topic. You might enjoy finding something.
Thanks for the comment. Yeah.
Thank you. Very inspiring talk. Very interesting. Just one thought that I had. Can this be a test to test availability of results, say, in medicine? You know that you can actually check, if a new product or new drug is being tested, that you then look at the results of these drugs, and if it doesn't fit this curve, you could say this is not a true result.
Yes. In fact, this has a practical application, and I have a patent being prepared to apply this to drug discovery research.
Just one more question.
So would you say that this is a formal process, or do you believe that there's actually, ultimately we believe physics sort of underlies everything. So is there simply an underlying physics that underlies a lot of phenomena we see? Is that what you're, is there a solid physics underneath this comparison, or is it a semantic?
I think there are two aspects of physics. Physics as reality, and physics as we model it. And so I have a category, which I call category of human knowledge, which starts with reality, phenomena, models. And the models can be physics can be viewed as models. And if it's isomorphic with reality, then reality is a physics. So we have to distinguish, I think, the two.